Hello friends, welcome to my YouTube channel. In this video, I will discuss about glycolysis. So let's start. Glycolysis involves the breakdown of glucose to pyruvate while using the free energy released in the process to synthesize ATP from ATP and inorganic phosphate. The 10 reaction sequences of the glycolysis is divided into two stages. First is energy investment step and another is energy recovery step. Let's read the requirement of glycolysis. The site is all cells in the body, subcellular site, cytosol, starting materials, glucose, end product, pyruvate in aerobic condition and lactate in anaerobic condition. Coenzyme, NADH, ATP steps are divided into two phases. So you can note down in your notebook. Overview of the glycolysis. Glycolysis is the first step in breakdown of glucose to extract energy for cellular metabolism. It is present nearby all living organisms. Operates in both aerobic and anaerobic condition, and pyruvate is the end product of aerobic glycolysis, and lactate is the end product of anaerobic glycolysis. Lactate is used for gluconeogenesis in liver, and anaerobic glycolysis is only pathway that provides energy for mature red blood cells. Okay, it is the total reaction step and looking very complicated, but uh, I will suggest you that just make the whole process stepwise. Then it will be very easy to remember. And let's start by the stepwise reaction. The steps, there are first five steps, that is called the preparatory phases, because we need ATP to preparation for the steps, and we get four ATP molecules after the five steps. After the five steps we get four ATP molecules. This step is called payoff stage, okay, or energy recovery phase. Okay, the first step, that is from start for glucose and end with glucose 6 phosphate. Here the enzyme is hexokinase, and then this first step of the glycolysis is catalyzed by hexokinase, an enzyme with a broad specificity that catalyzes the phosphorylation of the 6 carbon sugar. Here is the 6 carbon sugar and the phosphorylate there, and is by using the ATP, it break down the ATP and get a phosphate, and using this phosphate it phosphorylate the 6 carbon of the sugar, glucose 6 phosphate. Okay, so by using the hexokinase, and it can no longer live in the cell, it can no longer leave the cell, because the negative charge phosphate, this negative charge phosphate will not allow it to cross the hydrophobic interior of the plasma membrane. Okay, it was the first, and the next step is second step, uh, where the start from glucose 6 phosphate and end with fructose 6 phosphate. So here the enzyme is phosphorylase isomerase. This is an isomerase enzyme. So in the second step of the glycolysis, an isomerase converts glucose 6 phosphate into one of its isomers, fructose 6 phosphate. One of its isomer, fructose 6 phosphate. So an enzyme that catalyzes the conversion of the molecule into of its isomer, that is called an isomerase. So this function acted by this phosphohexose isomer. And here is the cofactor Mg2 plus that controls the negative charge of this phosphate group. So this change from the phosphoglucose to phosphofructose that allows the eventual split of the sugar into 2,3 carbon molecules. So let's see. This is the third step. From that, start from fructose 6 phosphate to and get fructose 1,6 phosphate by using phosphofructokinase 1. So the third step is the phosphorylation of fructose 6 phosphate, that is catalyzed by the enzyme phosphofructokinase 1. A second ATP molecule donates a high energy phosphate to fructose 6 phosphate, producing fructose 1,6 bisphosphate. In this pathway, phosphofructokinase is a rate limiting enzyme. Why it is called rate limiting enzyme? It's elaborate me. So it active. So it is active when the concentration of the ADP is high, and it is less active when the concentration of the ADP is low and the concentration of the ATP is high. So thus, if the sufficient ATP is there in the system, the pathway will slow down. So this is called rate limiting, uh, step, or this is the rate limiting enzyme. So next is, this is the fourth step. In the fourth step, that start from fructose 1,6 bisphosphate, and we get two 3 carbon molecule, that is dihydroxyacetone phosphate and glycerol 3 phosphate. Okay, so in the fourth step, of glycolysis, employ this enzyme, this is aldolase, and to cleave 1,6 bisphosphate into two 3 carbon molecules, that is dihydroxyacetone phosphate and glyceraldehyde 3 phosphate. Let's move to the fifth step. Okay, so it was the end step of the energy requiring step. Okay, here the dihydroxyacetone phosphate, and there is a glyceraldehyde 3 phosphate, and the reaction is the reversible reaction. So in the fifth step, an isomerase transforms the dihydroxyacetone phosphate into its isomer. This enzyme name is triose phosphate isomerase. So thus the pathway will continue with two molecules of a single isomer. So this pathway will continue with two molecules of a single isomer. So at this point in this pathway, there is a net investment of energy from two ATP molecules in the breakdown of one glucose molecule. It was the fifth step. Next, move to the second half of the glycolysis. It was the first half of the glycolysis. Next, move to the second half of the glycolysis. Okay, so let's move. It is the sixth step. The sixth step in glycolysis, this is a glyceraldehyde 3 phosphate, and we get 1,3 phosphoglycerate by using this enzyme glyceraldehyde 3 phosphodehydrogenase. The sixth step in the glycolysis oxidized it is oxidized and extracting the high energy electrons, which are picked up by the electron carrier NAD plus and producing NADH. The sugar is then phosphorylated by addition of a second phosphate group producing 1,3 phosphoglycerate. Here note that the second phosphate group doesn't require other ATP molecules. So here again there is a potential limiting factor for this pathway. The continuation of this reaction depends upon the availability of the oxidized form of electron carrier NAD+. Thus, the NADH, in order to keep this step going. So this NADH must be continuously oxidized back into NAD+. So the NADH must be continuously oxidized back into this NAD+, in order to keep this step going. So here the NADH is very important, has a very important role in this step, and it oxidizes back into NAD+, and this step is going on again and again. So 1,3 bisphosphoglycerate to 3-phosphoglycerate. Okay, so it is the seventh step. In the seventh step, catalyzed by the phosphoglycerate kinase, an enzyme that is named for this reverse reaction, but not here. But 1,3 phosphoglycerate donates a high energy phosphate to ADP. It donates its high energy phosphate to ADP and make ATP. This is substrate level phosphorylation. So here the carboxyl group of 1,3 phosphoglycerate, that this is a carboxyl group here, it oxidized to a carboxyl group, and a 3-phosphoglycerate is formed. And from here, that from here 1,3 phosphoglycerate, you can get 3-phosphoglycerate by oxidizing this carboxyl group. Okay, let's move to the eighth step. Is the eighth step, right? The enzyme is phosphoglycerate mutase. Phosphoglycerate mutase, and here the reversible reaction is seen here. So the enolase, not enolase. This is the eighth step. So eighth step is here the presence of the phosphoglycerate mutase. In this mutase, that in this eighth step, is very interesting. In this, the remaining phosphate group in the third step, and this is the remaining phosphate group in this in this third carbon, with third carbon, the remaining phosphate group This that moves to the third carbon to the second carbon, and this it moves to the third carbon to the second carbon, in producing 2-phosphoglycerate. So the reaction is 2,3-phosphoglycerate, 2-phosphoglycerate, and is vice versa, and the phosphoglycerate mutates by phosphoglycerate mutates and by using Mg2 plus. So this is mutase is an isomerase, is an another isomerase. Okay. Next, move to the ninth step. So here the enolase is present. So we make 2-phosphoglycerate to phosphoenol pyruvate. So PEP. So the enolase catalyzes the ninth step. This enzyme causes 2-phosphoglycerate to lose water from its structure. You know that it also act on this structure and lose water from the structure and make double bond here. So this is it is the ninth step and move to the ending step here. This is the 10th step. uh, the last step. It is the last step where this another substituted phosphorylation is happening here. It is because this high level, high energy phosphate this act on ADP and make ATP molecule again, and this is by using this this is by using this pyruvate kinase. Okay, the another is this is the end reaction, and where we get ATP again after the second step. Okay, so it was the ending step of the glycolysis. Now here you can see the whole step that I have elaborated here. I've elaborated here in the steps you can see, but it altogether look very complicated, but it is very simple if you try by with its chemical structure. That will be very easy to you for remember. And here another main point is we in this first step we see that from the hexokinase to this to fifth step we lost 2 ATP, we lose 2 ATP molecules, and we get 4 ATP molecules in after the fifth step. We get in the seventh step 2 ATP, and then another seventh step 2 ATP molecules. So more is we get 4 ATP molecules, but we lose 2 ATP molecules. So net ATP is 2 molecules. Okay, so our video has been ended here. Okay, but thanks for watching this video. If you like the video, please like and share with your friends and subscribe and hit the bell icon to get such kind of video.